everyone, and welcome to episode 56, the second episode in this series exploring the kingdom Plantaea. Last episode, I dived right into the wild lakes and ponds with their slimy algae crusts to explore the green algae ancestors of all plant life. Today, I'll be moving down the evolutionary lineage and exploring the organisms of the next divergence. These are the first simple land plants that evolved from the algae. These are the Bryophytes, the Liverworts, the Hornworts, and the mosses that cover so much of our planet. These Bryophytes include about 20,000 species, all of them small, non-vascular plants that compose the soft herbaceous carpeting of the forest floor. The first ancestral Bryophytes emerged from the algae mats sometime around 510 to 630 million years ago. Because the rest of the land plants would diverge from this Bryophyte lineage, the Bryophytes are like an intermediate grouping between the almost entirely aquatic green algae and the almost entirely terrestrial vascular land plants. They share qualities from both these groups, which is why the Bryophytes can live in both semi-aquatic and terrestrial environments. The Bryophytes share traits with the green algae, like having similar chloroplast structures and identical chlorophyll pigments. Although pretty much all plants have these same chlorophyll pigments, so I guess this is more an example of a trait in the founding population that persisted in virtually all of its descendants. More specifically to green algae and Bryophytes, the Bryophytes also depend on water for the transport of their sperm. They depend on water for reproduction. This critical trait really confirms their close relationship with the green algae, which also require water to reproduce. Some of the emergent traits that make the Bryophytes more able to tolerate life on dry land include a cuticle, or a thin layer of a waxy secretion that helps protect the outer layer of the plant from desiccating, from drying out in the sun and the dry air. They've also evolved to consolidate their cellular structure. Where algae are often just slimy clumps of filament and single cells, or ribbon-like growths of unvascularized tissues like kelp and seaweed, the Bryophytes have consolidated the important cells into distinct structures, like the gametangia organs that protect the gametes, now that they're being held up in the open air and aren't just easily dispersed around through the water. One of the most defining and important evolutionary adaptations that emerged in the Bryophytes was a stage of embryonic development. This is a period between conception and true waking life, where the young plant is developing on a chemical level. Where multicellular algae often grows in simple shapes with relatively low cellular differentiation, Bryophytes and the rest of the land plants are built with very differentiated tissues. To organize this differentiation and to lay the groundwork for a healthy body plan, the beginning of the plant's life goes through a period of embryogenesis. The zygote grows and develops internally before it germinates, wherein the cells are differentiating and getting organized so they can grow into a healthy, properly arranged, mature organism. Algae doesn't do this. They don't really need to. But the evolutionary emergence of this embryonic stage in early development laid the genetic and morphological groundwork for future plant evolution. The genetic and morphological architecture emerging in the Bryophytes is literally the material that subsequent plant generations would build off of to diverge into their own lineages, into the gymnosperms and the angiosperms and all their individual little families and genuses and species. Although, I shouldn't make it seem like the early evolution of the Bryophytes is set in stone. There's actually a lot of discussion over this issue, because genetic and proteomic studies keep yielding inconsistent results. Some posit that the Bryophytes and the ancestor of the rest of the vascular land plants evolved independently, and that the Bryophytes are a monophyletic sister clade to a monophyletic clade of vascular plants. There are studies that suggest that the Hornworts might not even be Bryophytes, that they might instead be more closely related to the vascular plants, and that the Bryophytes are really just mosses and liverworts. The more popular opinion, or the general consensus that seems to exist among most scientists, is that the Bryophytes are a paraphyletic group. This position argues that the Liverworts were the first Bryophytes to appear, from which diverged mosses, from which diverged Hornworts, from which diverged the rest of the vascular plants. In this way, the Bryophytes aren't monophyletic, because they don't include the vascular land plants, which share a common ancestor with all of the other Bryophytes. Alright, so let's take a deeper look at the Bryophytes, starting with the liverworts. The visible macroscopic structure that make up the liverwort's body is composed of haploid cells from the gametophyte generation. The liverworts are typically less than 10 centimeters tall, and anywhere from 2 to 20 millimeters wide, so they have dimensions that are measured in single-digit inches. They're often smaller or narrower than your thumb. Many species of liverwort have a growth form called a thallus, which is a multicellular organism whose tissues aren't organized into organs and/or they show little to no differentiation of specific structures. In a thallus of sargassum weeds, the plant just grows and branches continuously in a messy, tangled pile. There's no difference between the most recently growing branches or the middle branches or the oldest branches. It's all the same structures repeating over and over again. It's like it's got no ultimate goal or growth form, like a mature tree does. In liverworts, a thallus looks like a flat, slimy green sheet, with overlapping folds growing in an asymmetric, organic shape. Other liverworts have a more traditional, plant-like shape, with a central stalk or a stem structure that's dotted by pairs of leaves, with a clump or a node of specialized leaves at the top. Although, because these plants are non-vascular, there are no true veins running through their leaves or their stems, so when you hold a liverwort in your hand and look at it, it looks almost rubbery or like molded plastic. With regards to the rest of the plant kingdom, the liverworts are like pariahs. They're outcasts. After other bryophyte lineages had diverged from them, the liverworts experienced significant divergence in their own right, being the only extant land plants in the world whose sporophyte generation doesn't possess stomata, those little respiratory pores. This is because the sporophyte generation in the liverwort is tiny, almost microscopic, and very short-lived. When haploid sperm flows with the help of water into the archegonium to fertilize a haploid egg, the diploid embryo that results is retained on the archegonia. It's retained on the female gametophyte. The diploid sporophyte embryo grows into a mass of cells with a few rudimentary parts. There's a foot region that emerges and anchors the rest of the embryo to the surface of the archegonium, and it serves as a chemical corridor for nutrient transport between the mother plant and the offspring. There's a seta, which is like a thick connective fiber linking the foot to the rest of the embryo. And then there's the capsule, which is basically the rest of the embryo. The capsule is a globular balloon that is filled by all the new spores. When the seta grows, it extends and pushes the capsule up and out of the archegonium. Exposed to the air, these capsules can rupture, releasing their spores into the wind to be carried to whatever medium at whatever place where they land and germinate with their little rhizoids and become new gametophyte individuals. The germinated spore grows into a mass of filaments, which condenses into a lobular sheet or a chunk of biomass, which is the bulk of the liverwort's mature body. From the mature body of the gametophyte, the sex structures are lifted upwards on the ends of protruding stalks of tissue. In males, this structure is called the antheridia, which produces sperm cells. In females, the structure is the archegonia, and it produces and holds the eggs. In episode 53 on plant reproduction, I briefly talked about monoecious and dioecious plants. Well, liverworts can be both monoecious and dioecious too. Those liverworts that are monoecious have male and female sex organs on different branches, but on the same individual, while those plants that are dioecious have just one type of sex organ or the other on all of their branches. In all cases, the sperm of the liverworts have two flagella that they use to swim through the water, to get from their antheridium to the archegonia to fertilize the egg. Because some species of liverwort produce both male and female sex organs, it's entirely possible for them to self-fertilize, or to reproduce asexually. This often involves the use of structures called gemma cups, which are shallow, bowl or cup-like depressions in the plant's tissue. Inside of these gemma cups are disc-shaped wads of haploid tissue called gemmae. When raindrops fall into the gemma cup, they can splash and blast the gemmae outward, where they presumably land somewhere and germinate into a new clonal individual. Because this form of reproduction doesn't involve the genetic material of another individual, it's an asexual process. It's called fragmentation, because the parent individual is literally being fragmented or broken apart, and these fragments can survive and establish themselves to become new clonal individuals. Liverworts are most closely related to mosses, which share many traits like a dominant haploid gametophyte generation, a lack of vasculature, the use of spores to reproduce, and many other similar qualities. But there are some differences. So all the bryophytes, be they liverworts, moss, or hornworts, they all possess structures called rhizoids instead of roots. Where roots are thick and absorb nutrients, the rhizoids are thin filamentous structures that help anchor the bryophyte to its substrate. They help keep it upright, keep it in place. Unlike roots, rhizoid tissue isn't absorptive. It doesn't extract water or nutrients from the soil, or from whatever substrate it's growing on, be it rocks, tree bark, or acidic boreal soil. In liverworts and hornworts, each rhizoid is a single, huge, overly extended cell. But in mosses, the rhizoids are larger and thicker because they're multicellular. Where liverworts can grow in a thalloid structure with tangled masses of undifferentiated tissue, or in a foliose structure with an identifiable stem and leaves, the various moss species only grow in a foliose structure. The gametophyte generation of moss has a relatively simple morphology, including a stem with a few small leaves coming off of it. When you look at a big patch of moss, at thousands of little stems all at once, it looks fuzzy. This fuzzy texture comes from the stems being covered in leaves that are so small they might as well be hairs. The stems begin as a bud that grows out of the nodes in the rhizoid tissue. Because the rhizoid tissue runs horizontally under the surface of the soil, it can connect multiple shoots together. Thus, if you look at a little patch of moss and you see, for example, a dozen little shoots poking up, a dozen little fuzzy stalks of moss, this isn't necessarily 12 moss individuals. It's entirely possible, quite likely even, that the little patch of moss you're looking at is just one single individual with a dozen gametophyte shoots poking up into the air. Moss has reproductive mechanisms that are similar to liverworts. For example, they can grow gemea that can fragment off and generate a new individual. This new individual is a clone of the parent. The alternation of generations begins when a spore germinates and produces a mass of filaments or some kind of thallus, and from this intermediate structure the shoots of the gametophyte erupt upwards. A single spore can end up creating multiple little gametophyte shoots, which from our human perspective looks like a little tiny clump of fuzzy moss. The gametophores are thin stems, just a few millimeters tall, with numerous small little hair-like leaves. At the tip of the stalks are the sex organs. The female archegonia is nestled within the parachaeta, which are specialized leaves at the top of the gametophore that wrap around and protect the female sex organ. Just like the liverworts, the archegonia hold the eggs, and they have a column or a tube-like structure that sperm has to travel down to reach and fertilize the eggs. The male sex organs are the antheridia, and these are also surrounded by a protective layer of specialized leaves called the paragonia. Mosses are also monoecious or dioecious. Species of monoecious moss can reproduce asexually simply by floating some of their sperm a short distance to their eggs. Some of these moss species keep their antheridia, and thus their sperm, inside of little cup-shaped structures, so that when a raindrop lands in the cup, it'll splash sperm out several centimeters away in any direction. Sexual reproduction works in pretty much the same way. It just requires water as a matrix for the sperm to travel in, so that it can flow into the archegonia and fertilize the egg. Fertilization creates a diploid sporophyte zygote, which will grow into the mature sporophyte. In moss, this typically looks like a tall, thin stalk protruding from the short, wigglier, fuzzier moss gametophyte. At the top of the long sporophyte stalk is a capsule and a little cap to the capsule called an operculum. The capsule is filled with spores, and when the sporophyte matures, ages, and dries out, the operculum capsule degrades, and it falls off, and it releases the spores into the wind. In many species of moss, this degradation process creates an explosive release of the spores, spraying them some distance away from the parent. In sphagnum mosses, the kind that are covering the boreal forests and tundra of the world, they have shrinking capsules that create an increasing internal air pressure, and at a certain threshold, the pressure is too great, and it blasts the capsule open, launching all the spores out with it like a hand grenade. Like the liverworts, they also sometimes grow gemmacups and other similar structures that take advantage of the force of a splashing raindrop to propel their spores or their sperm. Remember that moss are bryophytes, and they require at least some water, at least a thin layer of water for their sperm to swim through. Just like liverworts, moss sperm also has 2 flagella that it uses to swim through the water. One of the largest clades of moss is the sphagnum genus. The sphagnum mosses exist all over the planet, but like I said a moment ago, they're especially concentrated in the northern hemisphere, where they grow on the bark of the trees in boreal forests, on the forest floor, and all across the vast expanses of tundra that characterize much of Canada, Alaska, Siberia, and Scandinavia. They're not exclusive to the northern hemisphere though, they exist in the southern hemisphere as well, in places like Argentina and Chile, New Zealand, and Tasmania. Where sphagnum moss grows, it tends to create peat moss, or peat bogs. This is an interesting process that starts with the quality of the moss's cells, and I'll work backwards from peat bog to moss cell to explain how it's all connected. So the organic material called peat, or turf, is a kind of soil composed of plant biomass at various stages of decomposition. It's generated as plants grow and die and grow and die, creating a rising carpet of vegetable matter building off of the decaying bodies of its forebears. When the plants decay, they give off CO2 and they release nitrogenous compounds, and the living plants on top of the decaying mass directly absorb these nutrients to fuel their own growth. In this way, the ecological relationship between the living plants on the surface of the peat and the decaying plants and the dead peat itself is almost self-sustaining. Obviously, carbon has to be brought into the system in the first place, and has to keep being brought in to replenish the carbon that settles in the decomposing plant tissue, but there is a fair degree of carbon recycling in just the upper layer of the peat. The majority of these decaying plants are sphagnum moss, which curls up and shrivels when it dies, and gets pressed below the young living moss, which is actively growing and reaching upwards to get at the sunlight. The living surface layer of moss on the peat kind of stomps down the dead and decaying moss below it, and over long periods of time, this builds up into peat soil that's several meters deep. All of this peat is very absorptive of water, extremely absorptive of water, so when it rains or when a river floods, the peat just sucks up a ton of it and holds it, and this makes the peat muddy. This accumulation of water in the soil can make the peatland very wet and soggy. And if this water is never released, say the peat was developing in a lowland area, if it never really dries out and more water just keeps getting added to the ecosystem through rain and flooding and whatnot, then the peatland can turn into a bog or a mire. These are generally treeless landscapes, like you would find in a grassland or the tundra, except the peat bogs have soil that's so saturated with water that there are ponds and lakes of standing water. This naturally feeds all manner of insects that come to breed and lay their eggs in standing water, like the mosquitoes in North America. The reason the peat soil is so absorptive in the first place is because the sphagnum mosses have large structural cells in their leaves, called hyaline cells. These little moss leaves use these hyaline cells for structural support, for mechanically filling out biomass, and they're amazingly effective at storing water. When sphagnum moss are growing in well-hydrated soil, they can absorb 16 to 26 times their dry mass in water. This makes their leaves look swollen, and it makes their branches look like they were some kind of gummy snack, like made of the same stuff that gummy bears are made of. The heavy internal turgor pressure that's plumping out their cells, it gives them this wet, plasticky, inflated look, kind of like a water balloon. I suppose that's a reasonable description, because the moss tissue is literally inflated with water, and the cell membranes and the cell walls are expanding outward to accommodate the water load, just like the balloon part of a water balloon. The sphagnum mosses retain this absorbency through life and into death, which is why the peat soil is so absorbent. The peat soil is made up of millions of dead little gametophyte shoots, and even though these are all dead and no longer growing, they still possess millions of hyaline cells that can still absorb and hold a huge amount of water. Alright, so before I talk about moss too long and run out of time, I'm going to move on to hornworts, which among all the bryophytes I've talked about, are the most closely related to the vascular land plants. The hornworts are the most closely related bryophyte to everything you typically consider a plant. Whether it be a pine tree or an orange tree, a blueberry bush or a tomato plant, a garden flower, a cactus, a fern, all of these things are connected evolutionarily to the Bryophytes through the Hornworts. Okay, so what are these things? What are Hornworts? Well, just like mosses and liverworts, the Hornworts are non-vascular. They undergo an alteration of generations with a dominant gametophyte generation, and they require water as a medium to facilitate their reproduction. Unlike mosses and liverworts, the Hornworts have just a single chloroplast in each of their cells. Where most plants typically have lots of chloroplasts in each cell, all busy working away conducting photosynthesis and producing energy to produce sugar, the single chloroplast of the Hornwort cell is integrated into a larger, more diverse chemical complex. This part chloroplast chemical superstructure produces and stores all of the food that the cell needs. In plants, this unique kind of arrangement is very uncommon and arguably inefficient. But it's common in numerous algae species, which are frequently composed of populations of just single cells. The alternation of generations in a hornwort works much like it does in the liverworts and the mosses. A spore lands somewhere and it germinates, with a single cell extending a tube-like projection that begins to divide at its tip. This leads to the development of the mature gametophyte, which has really varied morphology depending on the species you're looking at. The mature gametophyte can look like a continuous lumpy green carpet with thick leaf or lobe-like projections. They can look like tiny weeds or small shrubs, and others can look like weird lettuce without any veins in the leaves. From within their gametophyte body, whatever form it takes, the mature hornworts grow their sex organs and produce gametes. Just like liverworts and mosses, the hornworts have female archegonia and male antheridia, and various species can be either monoecious or dioecious. The archegonia and the antheridia are grown inside little cavities or pores in the surface of the gametophyte, which are only exposed to open air when the cells in the tissue above them die off and fall away. Basically, when the plant matures, these upper cells will die off and fall away, and they'll open up the sex organs. They can then release their sperm into the water, which will flow into the archegonium, and fertilization will occur. Upon fertilization, the zygote develops into the sporophyte, which grows into a stalk erupting out of the top of the gametophyte. This is the same case with mosses and liverworts. At the tip of this stalk is the bulbous capsule, which produces, stores, and then releases the sporophyte's spores. While bryophytes may not have had the most valuable or noticeable roles in the local ecology, they have played several important functions and roles in Earth's evolutionary history. For example, it's believed that early moss, spreading across the ground 470 million years ago, was responsible for extracting a lot of carbon dioxide from the atmosphere. As the moss grew and generated biomass, it absorbed CO2 out of the atmosphere. Also, as they grew, they dissolved the upper layers of the rocks that they were growing on. This organic weathering of the rocks did a few interesting things. First, by chemically altering the rocks were able to reabsorb carbon dioxide to recreate carbonate. And second, the process released mineral dust rich with calcium, magnesium, phosphorus, and iron, a whole lot of which got into the oceans. Because iron is a very limited resource in the oceans, when you see iron go into the water, you'll often see an explosion of growth. In most cases, seeding iron will feed algae, creating an algae bloom. The algae bloom involves the sudden and explosive growth of algae, which requires more CO2 to produce their biomass, and that directs the carbon towards biological sinks. This moss-fed process of altering rocks, producing sediment, and generating algal blooms is all very carbon-intensive, and it extracts a lot of carbon dioxide from the atmosphere. It's believed that enough carbon dioxide was extracted from the atmosphere over millions of years that the global temperature lowered and the world began going through a series of ice ages. Because mammals were heavily shaped over millions of years by the evolutionary pressures of a cooling climate, it's worth appreciating the subtle influence that moss has had on our own evolution. Well, that is about it for today's episode on the liverworts, the mosses, and the hornworts. I always find it fascinating how you can look at even the smallest, most seemingly mundane things, like some moss growing on a tree, and know that it's this amazingly complex multicellular structure with unique enzymes and chemical responses and ecological functions. Considering the ecological effects of these small organisms is also really fascinating, because it's just mind-boggling and surreal how such tiny things can have such a large, profound collective influence. This should be a good example for you to understand that because all life is interdependent on other life, and that all life influences all the other life in its habitat through ecological relationships, that even these small, seemingly innocuous plants have a role to play, that they serve some tiny but crucial purpose in a particular habitat, whether it be to supplement the diet of rodents, provide feeding opportunities for insect larvae, or by dying to accumulate water-absorptive peat that can eventually transform an entire habitat into a wetland. This interesting dynamic between tiny organism and the capacity to reshape a habitat is one of the fun things about life that makes biology so interesting. I hope you found this episode and this point about life as thought-provoking and as interesting as I did. If you're enjoying this series exploring the kingdom Plantae, then stick around for the next episode, which will explore the next two major lineages that emerged in the plant kingdom after the evolution of vascular tissue. First the Lycophytes, and then the ferns. And as always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.